For our project, the researchers assessed peanut allergy sensitization and oral tolerance in genetically diverse mouse strains. The onset of peanut allergy typically occurs during childhood and is a potentially fatal disease. Allergic sensitization to peanut and other food allergens occurs due to a failure to initiate or maintain oral tolerance. Additionally, non-oral routes of peanut exposure, including cutaneous and airway exposure, have been linked to allergic sensitization in clinical studies and mouse models. For prevention, the Learning Early About Peanut Allergy, or LEAP trial, is a seminal study that found that early dietary introduction of peanut reduced rates of peanut allergy. For genetic diversity, the researchers surveyed 12 collaborative cross, or CC, mouse strains. CC mice were specifically developed as a set of inbred mouse strains with genetically distinct backgrounds and have been used to establish mouse models of human diseases. The researchers asked the biological question, will early dietary introduction of peanut reduce peanut allergic sensitization and oral tolerance? And their hypothesis was yes, early dietary introduction to peanut will be efficient in reducing airway sensitization and inducing oral tolerance.
Assays and methods. So to perform this experiment, the researchers used 12 strands, or 12 different types of mice, and then they had 3 to 4 mice for each strand. The treatment mice were fed peanut butter every other day for 24 days, and then the control mice were just given regular mouse food the whole time. After the treatment mice had been fed the peanut butter for 12 days, so halfway through the experiment, they were sensitized to the airway by using LPS, which primes for allergy, and peanut protein. All of the mice, so the ones that were fed peanut butter and the ones that were just given regular mouse food, were treated using this inhalational model twice weekly for 2 weeks. Then the mice were challenged to peanut through an IP injection, and then after that, the researchers monitored their core body temperature every 15 minutes for an hour. What they were doing is they were looking for a drop in temperature that would indicate anaphylaxis, which would say or tell whether or not they were actually allergic to peanuts. And then to supplement the body temperature data, the researchers also took the blood of the mice before the study, before they did the inhalational treatment, and after all the treatments were finished. Then they took the serum from the blood and analyzed it using something called an ELISA, which is an assay that looks for peanut-specific antibodies, which were IgE and IG1, and those would also tell the researchers if the mice were peanut allergic.
For the results, 11 out of 12 strains that received regular chow developed peanut-specific IgE and or peanut-specific IgG1 after airway sensitization. While some of the strains that were fed regular chow experienced severe anaphylaxis, some of the other strains that were also fed the same diet were not affected at first. To test this, at a higher dose of peanut butter, they gave them 5 times higher than they had previously, and those strains then experienced anaphylaxis. When the 9 reactive strains were fed peanut butter before sensitization, none reacted. Most of the CC strains screened became tolerant or allergic depending on the initial peanut exposure. Overall, the mice that were fed peanut butter had less peanut-specific IgE, while the ones that were fed the chow diet had higher levels.
We determined that yes, the researchers interpreted the results correctly. They determined that allergic sensitization and oral tolerance to peanut can be achieved across a range of genetically diverse mice. Notably, the same strains that became allergic via airway sensitization were tolerized by feeding high doses of peanut butter before sensitization, suggesting that the order and route of peanut exposure are critical for determining the allergic fate. In summary, dietary peanut prevented significant induction of peanut-specific IgE or IgG1 in each case where the corresponding chow-fed strains had elevated peanut-specific IgE or IgG1. These results demonstrate that peanut feeding promotes oral tolerance in genetically diverse mice. These results suggest that the airway route of sensitization is plausible across genetic backgrounds and not strain or model-specific. 2 strains had severe anaphylaxis following a 0.5 milligram peanut protein challenge, while 3 strains did not experience anaphylaxis. Interestingly, each of these 5 strains generate peanut-specific IgE and IgG1, which suggests that their heightened or lacking reactivity to peanut may be due to upstream differences, including peanut-specific antibody affinity or mast cell or basophil number or reactivity. The non-reacting strains may have lower affinity antibodies or a higher threshold for peanut and might react if given a higher dose during the challenge. The varying responses across all 12 CC strains demonstrate the impact of genetic determinants on both oral tolerance and sensitization to peanut. Given that the majority of CC strains screened can become tolerant or allergic depending on the initial peanut exposure, it is clear that the order and route of peanut exposure are critical for distinguishing oral tolerance and sensitization. Regardless of genetic background, all CC strains fed peanut butter were resistant to peanut-induced anaphylaxis. These results further emphasize findings from the LEAP study that demonstrate the importance of early introduction of peanut for preventing allergy.
To start, the experiment first confirms that peanut allergy can form in a genetically diverse group of mice using inhalational or an airway model. I'd also like to add that there are very few models that sensitize through the airway like this. And then the study also showed that none of the mice that were fed peanut butter became peanut allergic. And because these mice received the same airway treatment as the mice who did become allergic on the regular mouse chow, it suggests that eating peanut butter before and during the airway sensitization can prevent peanut allergy. And these findings align with the clinical LEAP study. However, some strains of mice were not able to become peanut allergic. And this suggests that some components or some aspects of their genetic makeup prevent them from forming a peanut allergy, and this would need to be investigated further.
There are a couple of future ideas for experiments that could come from this experiment. This method of oral and airway testing could be used to test multiple different allergens and their potential for creating tolerance at a young age. But there could also be work done that identifies specific genetic factors and elements of the external exposome associated with oral tolerance and peanut allergy. More tests should be done to study the impact of environmental and genetic factors that affect peanut allergies.